Okay, guys, there's a lot to do today. Let's get started. So we're on to the next, we're actually going backwards a little bit. We're going back to chapter 5. We're looking at cell division. So this is probably something you've heard about in high school biology, but we're going to kind of reinforce it because it's very important. So first of all, what is cell division? It's the ability of organisms to reproduce. And this is one of the characteristics we talked about earlier that distinguishes living things from non-living things. And so the continuity of life is based on the reproduction of cells, so cell division. In other words, all cells come from previous cells. Cell division is an integral part of the cell cycle and begins first with nuclear division. So we'll talk about that in a second. So there's this thing called the cell cycle, which we'll go through in a second, and it's relatively simple in prokaryotes, and then it's more complex in eukaryotes. Let's take a look at prokaryotes first. So here's a picture of a bacterium that's dividing. So it's a mother cell turns into two daughter cells. Now, of course, they're not actually feminine, right? But we just use the term mother and daughter cell. So this is the type of asexual reproduction you see in unicellular organisms such as bacteria. And what you get when you have a division, you essentially get two new individuals that are genetically identical to the mother cell. So the daughter cells are genetically identical to the mother cell, and they also essentially divide up the cytoplasm and non-membrane bound organelles are divided up more or less equally. So most bacteria just have a single circular chromosome. So remember, that's one of the big differences between eukaryotes and prokaryotes. And eukaryotes, like us, we have linear DNA. So each piece of DNA has a start and a stop, and it's in a line. Whereas in bacteria, all their DNA is circular. And so for the majority of bacteria, all their genes are in one chromosome. So it's called a chromosome, but it's circular. There are, of course, examples of certain bacteria that will have two chromosomes, those two circular pieces of DNA, and some even have three. Okay, so there are exceptions to this idea that there's a single piece of DNA. So before cell division can occur in a bacterium, what has to happen is you need to replicate the DNA. So the DNA is circular, and so we've talked about this before. Deoxyribonucleic acid, it actually consists of two strands of DNA. You guys remember that? So it's the same thing in humans. In us, it's no different. We have two strands of DNA. And basically, so what we need to do is, if say this is the mother cell and this is the DNA of the mother, we have to divide this DNA so that we have essentially make copies and the daughters get these copies. So how does this occur? Well, What happens is you need to actually open up the DNA. Now, how does that occur? Well, remember we spent a long time on different types of chemical bonds. What kind of bonds are there between the two single strands of DNA? So DNA forms a double helix, right? Meaning it's a coiled structure with two single strands intertwined. Now, in the middle between the two strands of DNA, what kind of a chemical bond is it? Is it a covalent bond, polar covalent bond, hydrogen bond, ionic bond? Yeah, hydrogen bond, right? That's what you're going to say too, right? Yeah, so it's a hydrogen bond, right? So remember, those are really weak bonds. They're not nearly as strong as a covalent bond. And that actually makes a lot of sense, because even when you're not replicating DNA, the DNA has to open up a little bit so that you can go in and you can use enzymes to make an RNA, to essentially do transcription, which is to make an RNA, messenger RNA from the DNA, okay? And this is also very important when it comes to replicating the DNA. So what we need to have, we have enzymes that will open up the DNA. So it'll open it up. And then we have other enzymes that will start to bring in complementary base pairs and start to add them in. And they'll start to make new DNA, okay? And that can happen on this piece also. So it'll start here. And it usually travels like a wave as it's adding more and more bits of DNA. So the new DNA that's being added is in red. A little bit later, these things, as their DNA is being replicated, it's going to look more like, let me see if you get the picture right. It looks more like this, heart-shaped, and so we're continuing to synthesize new DNA. So there are enzymes that are doing all this for the bacterial cell. And eventually you get to the point where you completely replicated the DNA. So now you've gone all the way around this circular piece of DNA. And you have the original DNA in blue. And you have what's called the complementary strand of DNA, which is shown in red here. So that's all newly synthesized DNA. And of course, you have two of these now, right, by the end of this process. So the synthesis of new DNA has to occur before the cells divide, right? So, quite a bit before these cells divide, you have the new DNA. Actually, you can see this in this picture here. You have the original DNA, you have the replication of DNA or DNA synthesis phase here. Now we have two copies essentially of the DNA. So the blue and the red. The cell then elongates, and the DNA will be pulled, one copy of the DNA will be pulled, the chromosome will be pulled towards one end, the other towards the other end. And then you'll start to form this cell wall between the two halves. And then the plasma membrane will pinch off and so forth. And now you have two daughter cells. And the daughter cells are absolutely perfectly identical to the original mother cell. Okay. So this is referred to as asexual reproduction, right? So this is asexual reproduction. Okay. So the advantage of asexual reproduction is you don't have to find a partner. You could just be a single bacterium in a gigantic petri dish. And within a few hours, you got millions and millions of bacteria, all that are essentially genetic clones of the first one, right? So it's very rapid, it's very efficient. If you think about with sexual reproduction, half the population actually doesn't directly generate the next generation, right? Just the females do, right? The males just contribute some sperm, the sperm, which has genetic information. So asexual reproduction is very efficient. Happens, you can have a rapid increase in population. The big disadvantage. What's the big disadvantage with asexual reproduction? Can anyone guess? Go ahead.
It's not genetically diverse.
Right. With sexual reproduction, when you make offspring with a male and a female, there's a tremendous amount of shuffling of genes that happens, right? So you essentially get a new combinations. And sometimes those new combinations are extremely advantageous. Sometimes they're also disadvantageous, right? But so it's a bit risky, but overall you get a lot of diversity in the next generation. Whereas here you don't get that. You're basically just making essentially identical copies of the original. So asexual reproduction does occur in eukaryotic organisms. So for example, lots of plants do this, like strawberries will send off little shoots and essentially grow up a new little strawberry bush right next to, little strawberry plant, right? So it has its advantages and so forth. So here we go. Here's a nice picture of what it looks like. Here's an electron micrograph. It's a transmission electron micrograph. And you can see the nucleoid region. So what's the nucleoid region? This is the region where the DNA is. So remember, there is no membrane-bound nucleus in a bacterium. And then the DNA has been replicated and the cell's just about to divide. Okay. So what about us? Do we do something similar? Do we have to divide our cells also in a similar manner? And the answer is yes. So you're a multicellular eukaryotic organism, and you need to divide your cells and make essentially perfect copies, just like this one just did, because you started out as a single cell, right? So there was a sperm that got together with an unfertilized egg, and they made a zygote, right? So that was a single cell. So you actually started life as a single cell. That single cell divided, and then there were two cells. Those two cells divided, and there were four cells became 8, 16, 32, so forth, right? Then you have an embryo, and eventually that embryo transitioned to being a fetus, right? But all the time, all your cells are essentially making perfect copies of the original cells. So every single cell in your body, I mean, there are some exceptions. For example, red blood cells don't have a nucleus, but in general, almost every cell in your body has the full complement of DNA that was in that original zygote. Okay, so basically what I've been talking about is growth. You need to reproduce your cells. Your cells need to divide for growth. You need to do this for development from fertilized egg. And then even once you finish growing, once you're an adult, you need to repair your body. So your body is basically being damaged all the time, and also cells wear out. So an example would be white blood cells in your blood. The majority of them do not stick around for very long. They stick around for maybe a week or two. There's some exceptions. There are certain memory cells in your immune system that will remember when you're infected for numbers of years. They will stick around for many years. But the vast majority of white blood cells are so active that they get worn out, and within a week or two, they're dead, right? So you need to keep generating new white blood cells, which are involved in immune function. What about your skin? You're probably aware that the outer layer of your skin is dead, right? It's keratinized, meaning it has a lot of keratin. Beneath this keratinized layer of dead cells that is constantly coming off, you have cells in the epidermis that are essentially constantly dividing very quickly and generating new skin for you, right? Otherwise, you'd run out of skin within a week or two, right? It would be pretty horrible, right? There are other places where you have constant repair and growth. So for example, whenever you exercise, particularly intensely, you're going to damage your muscles just a little bit, so you have to repair them. Your alimentary tract, the gastrointestinal tract, which is the tube used for feeding, that is subjected to a lot of wear and tear. There's enzymes, there's acidity in the stomach, and so forth, right? So you're constantly repairing and regenerating the lining of the gastrointestinal tract. So occasionally, why is this important? Well, obviously we need to grow, we need to develop. That's the first thing. Repair is really a big topic in medicine. Why? Well, because sometimes there are problems where the genes aren't copied perfectly, and that can lead to things like cancer. Okay. So it's not surprising that skin cancer is a big problem in humans. Why is that? Well, because the skin is exposed to the elements and also to UV rays, but also because skin cells are constantly dividing all the time. as you get older, you know what a colonoscopy is? Okay. Once you get older, you're going to get the talk from your doctor. He's going to say, oh, it's time for you to schedule that colonoscopy. So they're going to look at your large colon and so forth. Why is that an area that's of such concern that where people get cancer? Well, because again, the cells are rapidly dividing all the time because of the wear and tear of the GI tract, right? And so it happens that even without toxins and things like that, there will be some problems occasionally, and cancerous cells will develop. And that's the sort of thing you want to figure out early on, because you can remove precancerous cells, which are polyps within the large intestine or colon. Okay, so again, this is why this is all very important. We need to understand this because we all developed, we all grew, and we also repair our bodies constantly. Okay. So let's talk about eukaryotic organisms. Well, first of all, we have a lot more genetic material in the form of DNA than prokaryotic cells. So I put the numbers down. You don't have to know the numbers here, but it's just sort of interesting to know. We have roughly 10 times more genes than a bacterium, but actually we have 100 times more DNA. So that's kind of an interesting story. We don't know exactly what's going on there. It seems like there are large stretches of DNA that don't code for protein, for example, in humans and other eukaryotes like plants and so forth. That used to just be called junk DNA. We're now finding out that some of it may be junk, some of it is actually important regulatory sequences and things like that. So that's still an ongoing story. Exactly why we have so much DNA is still being worked on by scientists. What else? Their DNA is also, so our DNA is also stored and packaged differently in the cell. So I already talked about the fact that our DNA is linear rather than being in a circle. Well, we'll talk about how it's packaged also. Okay, eukaryotic chromosomes are organized in the nucleus. So first of all, we have this membrane-bound organelle called the nucleus. It actually has a double membrane, right, which we've talked about before. All the DNA in a cell constitute the cell's genome. So we have DNA in the nucleus, which you can see here. But don't forget, we also have the mitochondria. They have their own DNA. And that DNA actually looks like bacterial DNA. So it's circular in form. If you look in a plant, they have mitochondria also with their own DNA. And they also have chloroplast. And the chloroplast DNA is also circular in form, right? So that's really intriguing. Why do they have circular DNA and not linear DNA like the DNA we have in a nucleus? We talked about this. Do you guys remember the endosymbiosis theory? So the idea is that our cells are actually archaean cells. Archaea are very simple cells that look like bacteria, but they're sort of a different lineage than the bacteria. So the idea is that this ancient archaean cell actually engulfed bacteria, some which became mitochondria and other bacteria that could do photosynthesis that became chloroplast. So that's why the DNA and the ribosomes and so forth within, they have their own ribosomes within mitochondria, and chloroplasts look very much like bacterial DNA and bacterial ribosomes. Okay, so our DNA that's in the nucleus is actually organized into chromosomes. So let's talk about this. So how do we pack? So we have a lot of DNA, right? Way more than a bacterial cell, 100 times more. So this is a problem. How are we going to package it? So DNA is going to combine with protein to form something called chromatin. And then that chromatin together with the DNA will form the chromosome. So chromosomes are composed of chromatin, a complex of DNA and histone protein. So I've actually got a picture of this down here. So we have DNA, and what it's doing is it's wrapping around a group of 8 histone proteins. And then there's a bit more DNA here. And then the DNA strand wraps around another histone protein, a bit of more DNA, and then it wraps around again, okay? So why are we doing this? Well, if you had a bunch of string in the house and you didn't quite know what to do with it, you wanted to actually keep it useful, right? You might wrap it around, find something in the house and just wrap it around it as a way of sort of storing it. And so what we're doing here, what your body is doing essentially is storing the DNA in a more compact fashion than it would be if it was just straight DNA without any associated protein. Okay. So, oh, these numbers you don't have to know. It's a tremendous number of nucleotides. Remember what a nucleotide is, right? That's the basic subunit of the DNA. The DNA is a nucleic acid. So DNA is coiled around these histone proteins in order to allow it to fit in a small space, despite being extremely long. So, and this is the rule. You don't have to, again, it doesn't matter. These exact numbers don't matter, but it's wrapped around these 8 histone proteins. That's what you're seeing here. And the wrapping is kind of curious. It's about 1.65. Again, you don't have to know that number exactly, but the main idea is that the DNA is wrapping around these things, and that allows the DNA to be more compact. So that's now called chromatin when you have DNA and proteins together. Okay, so here's a picture. We have some DNA here. It's the double helix. Here's the histone proteins. They can self-assemble into this complex. And then the DNA complexes mean it just combines with the histone. And then here we go. The histones can then assemble together. They're actually able to then be more compact. Okay. Oh, and this thing here, I should have mentioned this earlier. When you have, did I mention it earlier? Yeah, nucleosome is made of DNA wrapped around 8 histone proteins. So each of these little beads, it's often thought of like beads on a string. The histone together with the DNA is called the nucleosome. Okay. So the nucleosomes then assemble into the chromosome, which is seen here, and they're even more. So they sort of naturally want to assemble into a nice compact fashion if they're allowed to. But there are also additional proteins that help with this packing. So there's a lot of energy going. Oh, here's the beads in the string. See these little nucleosomes here? And then you can see how this stuff can become incredibly compact. And so when we see it in this form, this is your typical chromosome picture, right? It's incredibly compacted. And why do we need, so your DNA is normally not this compact, right? It's only when the cell is about to divide that it's essentially like if you were going to move from one house to another, right? You could be a little bit messy once you're living in a house, but if you're about to move, you have to start organizing everything into boxes and so forth for that move, right? So that's what's happening here. The cell is organized, getting itself organized just before a move, which is in this sense, it's going to be, in this case, it's going to be dividing into 2. Okay, so let's talk more about eukaryotic chromosomes. So there's a company out there. Has everyone heard of 23andMe? Okay, they're like advertising all the time that you can order, I don't know, you give them a sample of your DNA, and then they will tell you like where you came from, things like that, right? So that's actually kind of helpful because basically humans have 23 pairs of chromosomes, right? And that means, well, 23 pairs means you have 46 chromosomes altogether, right? 23 times 2. So in each pair, you have a pair of what are called homologous chromosomes, or also called homologs. What are they? Well, here's a picture done by an artist. So schematic sense, well, here's what it would look like. You would have a chromosome and another chromosome, and they're going to be the same length. They're going to have similar genes, okay? We'll talk about that much later, but you'll have similar genes at similar loci, locations on the chromosome. Okay. All right. So, oh, the other thing is for each pair, you're going to get, you got one, for each homologous pair that you have, one chromosome came from your father and one came from your mother. So for example, here, this purple one on this person might have come from their father, and this green one might have come from their mother, okay? Cells with pairs of chromosomes are called diploid or 2N, okay? So this is terminology we need to know. So most of your cells contain the diploid number of chromosomes, or 2N number of chromosomes, which is 46, okay? So diploid is from the Greek word for double, and oid simply means having the form or resemblance to something. So it's diploid or double. So the vast majority of cells in your body are all diploid, right? If I looked in your body, 99.999% are all diploid. But there are some cells in your body that only have the haploid number. Cells, they only have one copy of the chromosome. These are called haploid. So So they would, in place of having two, they would either, for a particular chromosome pair, they would just have one out of the pair. So they might have the chromosome from the father, or they might have the one from the mother. Can anyone guess what cells in your body might be haploid? Yeah, exactly. So the germline cells, right? So they're cells that are going to make the gametes, right? Which are the sex cells that are involved in sexual reproduction, right? So they're going to be haploid, right? So that's a very small minority of cells within your body that will only have one copy. We're going to come back to that. So these are called haploid. And the way we sort of say this in a short way is we just say they have the N number of chromosomes. Diploid, that's the double number, which is 2N, and that's what most of your cells have. Okay, so this brings us to a karyotype. So karyotype is an arrangement of a cell's chromosomes, and so this is done for medical reasons. When you have a couple, for example, and they suspect they may have some sort of genetic disease in their lineage or something like that, then what you would do is take a blood sample from the person. You'd separate out the white blood cells. These are living cells. You would find some that are, you put them in culture. White cells can be cultured pretty easily. Look through a microscope. Wait till a point where you see some white blood cells that are dividing. So they're going to have to do replication. So there's a point, again, when you have to essentially pack your DNA up. It's going to be right around what's called metaphase, which we're going to get to later, right? But there's a point called metaphase in which the DNA is really maximally condensed. And you would then add a drug that will stop, stop the process, right? Essentially going to poison things. And then you would also add some stains. And then you get these really brightly colored chromosomes. And then what you do, and this is kind of done with a computer, but you take these pictures and then you have to, because everything, of course, is oriented in a crazy fashion. You have to sort of play around with it a bit. But then you start to organize everything and you match up the pairs of chromosomes, right? So again, one would come from the mother, one would come from the father. And then basically you then put them all in order. So here, for example, you have chromosome number 1. So everyone has a chromosome number 1. Note that this is the longest. If you look at this, this is the longest one, right? So it has the most genes, and there's 2, 3, 4. Again, here they're showing a homologous pair of chromosome 4. So your chromosomes are all numbered all the way up to 22, right? And note that you see how these chromosomes are getting shorter and shorter and shorter. That's just, the way they've been arranged. Historically, that's how they did it. And then, but I said there's 23 pairs of chromosomes. So there's one last pair here. What's this last pair here that I'm pointing at? What's this about? We have an X and a Y. Yeah, so these are going to determine the sex of the individual, right? So if you have an X and a Y, are you male or female? Male, right, yeah. So what would it be if this individual was a female? What would you see there? XX, right, exactly. Okay, so this is a very useful technique. It's a way of looking for possible genetic problems. What can happen, and this again is not a topic that will come much later, but occasionally get inversions where a piece of DNA might be there, but it's flipped around, it's actually backwards and things like that. So that would be a genetic abnormality. Okay, so the 22 non-sex chromosomes are called the autosomes, okay? These contain genes that control traits other than sex. And then there's this one pair of chromosomes that contain genes that control sex-specific traits. So notice that the X and the Y are very different. X is much bigger than the Y, okay? And then there's kind of a cute little video. So I put the link there. So you can, I posted this, of course, on Blackboard. So if you want, you can take a look at that video there. Okay, so let's talk about the eukaryotic cell cycle. So what are the things that have to happen? Well, we need to copy all the DNA, which is packaged, of course, in chromosomes. We're going to have to organize all the chromosomes. So we're going to have originals and copies. And then we're going to have to do cell division, and we have to ensure that each daughter cell gets one set of each of the copied homologous chromosome pairs. Okay. So here's the phases of the cell cycle. There's interphase. So our cells most of our cells spend at least 90% of the time in interphase. Okay. And then 10% is the mitotic phase, which is the actual division. The mitotic phase includes both mitosis, which is actually division of the nucleus, and it also the cytokinesis, which is the division of the cytoplasm. So let's go back to interphase. So interphase, it may look like the cell's not doing a lot, but it's actually doing a tremendous amount. So there's the G1 phase. The G stands for gap, so it's the gap1 phase. This is the stage before DNA synthesis. So a lot of preparations are going on there. If you want to synthesize DNA, what do you need to do synthesis of DNA? What's DNA made out of? Yeah, nucleic acids, nucleotides, right? So you need to start building nucleotides and things like that. So you need to start making all the components that you will need in the next phase, which is the S phase. S stands for synthesis, so DNA. You have DNA synthesis, or also called DNA replication. Those are identical statements. And then finally, we have the G2 phase, the gap2 phase. This is the stage after DNA synthesis. So the G2 phase, we're going to start preparing. We're preparing for the mitotic phase. So we'll see that in that phase, a lot of enzymes will be made that are involved in the mitotic phase, and also things like microtubules and things like that will be made. So there's a lot of activity going on in those phases. Then we have mitosis, the actual division of the nucleus, and the cytokinesis, which is the division of the cytoplasm. So let's look at this term cytokinesis. It's a fairly old word. It's cyto means cell, which we've seen that before. Kinesis just means motion or movement. So it essentially means motion or movement of the cell. Okay, let's look at this picture here. So we've got all the phases here. You can see it's a cycle. It goes around and around. So the majority of this, 90% of the time or more, is spent in interphase. Here in this chart here, in this table, we have G1, S, and 2, and then the mitotic phase. And they're showing this in hours. So we have 5 plus 7, that's 12, plus 3 is 15 hours spent in interphase in this particular case. And then 1 hour in the mitosis. Okay, so more than 90% is spent in interphase for this particular cell. So here's the G1. So let's give you an idea of how long these phases are. The G1 phase is relatively long. Then there's the S phase and the G2 phase. Then we have the mitotic phase, the M phase. So first we have nuclear division. That's the DNA synthesis. The DNA synthesis is over here. Here we have nuclear division, where we divide the nucleus first. And then we have the cytokinesis, which is at the very end, split it into two separate cells. And then if we look at mitosis, which takes roughly an hour in a typical human cell, it can be divided into a number of minutes for different phases. So there's prophase, which is 36 minutes roughly, metaphase, which is relatively quick, 3 minutes, anaphase, 3 minutes, and then telophase, which is 18 minutes. So we're going to talk about all those phases in a second. So about an hour. I mean, these are just very rough numbers, right? That just gives you an idea of the timing of this. So G1 is the gap1 phase. The cell double. Here's some of the things it has to do, right, for preparation. It needs to double its organelles. It's going to accumulate lots of materials for DNA synthesis. So the cell is going to make a variety of proteins that are needed for DNA replication. Also the nucleotides itself, which we mentioned earlier. There is something called the G0 phase. What is that? Well, this occurs in some cells. They're going to pause to carry out normal functions, but don't prepare to divide. So what kind of cells do that? Well, throughout your body as an adult, you're going to have tissue-specific stem cells. So these are called adult stem cells. And they're just kind of stuck in the G0 phase. So they're just sort of living, doing their thing, waiting and waiting. So it's a reversible G0 phase, because they could go back to G1 phase and start getting ready to replicate. So if you look at skeletal muscle, for example, if you damage your skeletal muscle, you can actually regrow some new skeletal muscle, because what you have are these cells called satellite cells that will essentially be able to regrow more muscle for you. okay? Okay, let's go on to the S phase. So we have DNA replication that occurs. After each DNA replication, each chromosome has gone from one DNA molecule called chromatid to two DNA molecules called sister chromatids. So a picture is shown here. Let me draw it on the board. So, yeah, I'm just going to do the blue one here. So we have a chromosome here in blue. So there it is. And we're going to need to double our DNA. So what we're going to do is during the synthesis phase is we'll make a copy of this DNA. So it's essentially a perfect copy of it. So maybe this was the original, maybe this one came from your father, right? And each homologous pair of chromosomes, which is shown here, you have one, the blue one that came from your father, you have the green one that came from your mother. So what I'm showing you on the board here is just the one, the blue one that came from your father. It's been replicated. And now they're called sister chromatids, okay? And this is all in preparation for the fact that when you divide, you're going to need essentially double the amount of DNA. Does that make sense? You have to double up your DNA first before you can divide. So now the next phase, after the S phase, is the G2 phase. The cell synthesizes enzymes and proteins for cell division. What do you need for cell division? Well, we're going to talk a lot about microtubules, which are required for mitosis. Okay, what are microtubules? I think I talked about it briefly previous in this class. Does anyone remember what a microtubule is? Yeah, so a microtubule looks like this. It's made out of, there's these globular proteins, and they form a tube like this, and then my art skills are not great, but basically they're going to make this cylindrical tube that can be very, very long, formed of all these globular proteins called tubulin, right? So that's a microtubule. There are intermediate filaments, and then there are microfilaments, which are actin. Also, so well, these will all be important. Particularly, we're going to talk about microtubules and the microfilaments in a second when we talk about further about mitosis. Okay, here's a picture. Again, you might have gotten a blue chromosome. This might be chromosome number 4, for example, in your body. Before your cell, let's say it's a skin cell, is going to divide. Eventually, it needs to go through the S phase. And what it's done is it's made a sister chromatid. Okay? And the sister chromatids are often held together by what's called the centromere, okay? Which is a bit of protein. And then, what it's showing you is after through this process of cell division, we don't want to, your daughter cells can't have double the chromosome, so they have to genetic material, so they need to, we're going to divide, you'll see within mitosis, we'll divide up the genetic material, so go equally to both daughter cells. Okay, so here's the process. So DNA replication makes identical copies of homologous chromosomes. They're called sister chromatids. To organize the replicated DNA, the sister chromatids are held together by a structure around the narrow waist called the centromere. Okay, and you can see a centromere here on the homologous chromosomes. During replication, you still have the centromere here, and now it's holding the sister chromatids together. So, okay, one thing I got to point out here is, so this is, I think, confusing to people. So here we have a pair of homologous chromosomes. Each one of these things is called as a chromosome, right? So this blue one here is a chromosome, right? And this green one here, that's a chromosome. Now, after DNA replication or DNA synthesis, which is the same thing, you now have sister chromatids. Each of these, this is still considered a chromosome, okay? It's one piece of DNA because it's stuck together by the centromere. So one way of, I realize it's confusing, but one way of counting this is you just count the number of centromeres, okay? So let's go over here. How many chromosomes do I have here? Well, 2, because I have this centromere on the blue one and I have a centromere on the green one, right? That's 2 chromosomes. How many chromosomes do I have over here? Well, I have 1 centromere here and I have another one here, right? And so I actually only have 2 chromosomes here, okay? I think that can be very easily confusing because it looks like there is in fact double the amount of genetic information here, right? But it's we only count that as 2 chromosomes because they're stuck together at the centromere. Not working. Huh. It's not working. No digital.
During G1 or GAP1 phase, all the organelles and cytoplasm. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or GAP1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or GAP2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase and a very short period of time actually dividing, a process called mitosis. The replication.
Okay, okay. Okay, so eukaryotic cells undergo 2 different mechanisms to divide up the DNA. We're going to talk about mitosis first because it's simpler, and then we'll tackle meiosis later. So mitosis is a cell division mechanism that occurs in non-reproductive cells. So it's this asexual reproduction, which we need again for you to have developed from a zygote, a single cell, into an embryo, that embryo into a fetus. And then obviously, since you were the first day you were born, you've grown a bit, right? So that's all through asexual reproduction or mitosis. And also the cellular repair I was talking about, that's all mitosis. So these cells are called, so most of your cells are called somatic cells or body cells. So the term soma simply means body. So that's where that comes from. Meiosis is a cell division mechanism that occurs when cells participate in sexual reproduction. So we're going to do that second. We'll get to that later. These cells are called germ cells or gametes. Okay. So let's see here. So they're basically for mitosis, there are 2 distinct stages. Mitosis is division of the nucleus. It follows interphase. The sister chromatids separate into daughter chromosomes. And then these daughter chromosomes are distributed to the 2 daughter nuclei. And then after mitosis, we have cytokinesis, which is division of the cytoplasm. The 2 daughter cells result from the division are identical to the mother cell. So let's look at this. So we have mitosis, division of nuclear contents, in which the chromosome number stays constant. So we start off with 1 diploid cell, which is a 2N cell, and we're going to have 2 diploid daughter cells. So again, we call this first one the mother cell, and the 2 other cells daughter cells. They're not actually feminine, right? But we just use that terminology. DNA replication occurs before mitosis, producing duplicated chromosomes. So let's take a look at this. So we have, first of all, we just have our cell in interphase. Now our cells have 46 chromosomes, right? 23 pairs. If we look at this individual, how many chromosomes does it have? It has 4, right? So it's some other eukaryotic cell. It's not a human eukaryotic cell, but it's for illustrative purposes, we're using a simpler cell than ours. So it's 2N numbers, diploid numbers 2. This means that if there's 4 chromosomes, there are actually just 2 pairs, right? And we can actually see that here. What do we see when we look at this? We see, you see how they made them different lengths to help us out here? So let's do that. Let me redraw this on the board. So we have two short ones, right? So we have a short blue one and we have a short red one. Okay, so that's one pair. And then we have a long blue one and a long red one. Okay. So these are homologous pairs. We have two pairs here, so 4 chromosomes altogether. The blue one would have come from the father and the red one from the mother. Okay. Now, what's happened? Well, we're going to need to do DNA replication during interphase. So what's going to happen? We're going to double up all the DNA. So let's go ahead and do that. So we're going to double up all the DNA. So everything's like this. And I need to do 2 more of these guys. And what are they? How are they attached? They may be attached by what's called a centromere, which is a bit of protein. Right, something like this. Okay. So how many chromosomes do I have? 1, 2, 3, 4, right? If I count the centromeres, I still only have 4 chromosomes. But you can see that I've essentially doubled the amount of genetic material, which is what's going to be necessary to go to the next phase. So that was all occurring during interphase. We've duplicated the chromosomes, so we have sister chromatids. Now, when we go to mitosis, what we need to do, and this has to be done in a very organized manner, is we essentially need to get back to what the original cell had, right? Which means that we're going to take these and divide them up. We say, okay, well, one blue short one's going to go to one daughter cell. A blue short chromosome has to go to the other daughter cell. A red one's going to go to one cell over here. A red short one's going to go to the other chromosome. So not the other chromosome, to the other cell. So what we need a long blue one down here. And we need a long red. So we're dividing it all up. And in the end, what we have is we've divided up the DNA. And we're back to the original case, right? We have a homologous pair for the short ones, homologous pair for the long chromosome, homologous pair here for the short chromosome, homologous pair here for the long. Okay? So that takes a lot of organization. And so what we're going to go through next is to figure out how a cell can do that, how it can do all this organization to do that properly. Because things could go wrong, right? In fact, they do go wrong, which is something we'll talk about not now, but a little bit later. We'll talk about diseases in which this process does not go well. Okay, so mitosis follows interphase, and it's a continuous process, but we've divided it up into distinct phases to make it a little bit easier to understand this: prophase, metaphase, anaphase, and telophase. And sometimes prophase is actually divided into a number of stages because a lot of things happen in prophase. So sometimes we say prophase and then prometaphase, which is this intermediate step. Okay, we've looked at the numbers here before. Let's take a quick look at this video here.
The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells.
Okay, so there's there's a very good video, but there's a lot in there. So let's look at each phase really carefully here. So we have prophase. So here are the things that happen. The first thing that happens is nuclear membrane starts to fragment. You guys remember what the nucleolus is? What does the nucleolus do? It's found within the nucleus. There's a lot of confusion on this on the left. What's the difference between, okay, you know what a nucleus is, right? That contains the DNA. That's the double membrane structure. What's the nucleolus? No. Yeah, so the nucleolus is a dark region within the nucleus. And that's where RNA subunits are formed together with some protein. And those will become they're ribosomal subunits, right? So it's where the ribosomes are actually mostly RNA with a bit of protein, right? So the subunits are formed in the nucleolus, they're exported out through the nuclear pores, and then they become actually full-fledged ribosomes out in the cytoplasm of the cell. Okay, so that's what the nucleolus is. So that's all the stuff you don't really need. We're not going to be doing all this stuff right now, right? So the nucleolus starts to disappear. We don't need the nuclear membrane anymore. Now there's a lot of things, just some confusing terminology. Let's just tackle this head on. Yeah, so these things are called the protein that holds the sister chromatids together. That's called the centromere, okay? So this thing, there's another thing that has a very similar name. It's called the centrosome, okay? But they're completely different, right? What is the centrosome? Well, we saw this in the video. Around the cell, every cell has these centrioles. Every animal cell has these. What is the centriole? Well, it's essentially made out of these microtubules. So sort of a central point in which microtubules can originate from, right? And they're sort of arranged always perpendicular like this. The centrioles are found within a bigger structure called the centrosome, which again is showing here like these microtubules jutting out from it, almost like looking like a star, right? So centromere, again, holds together the sister chromatids. A centrosome is the bigger structure, and at the heart of the centrosome are the centrioles, okay? So this is very important for the generating animal cells. It's incredibly important. You have the centrosome with the centrioles within it. This is the sort of center for making a spindle. So we're going to make this football-shaped spindle structure, spindle apparatus. And so animal cells need that. So it turns out most plant cells, like flowering plant cells and so forth, do not have either centrioles or a centrosome at all. Some do, like there's some plants that do, but the majority do not. They do have a spindle, but they organize it slightly differently. Okay, we'll get to that later. So what else is going to happen in prophase? Chromatin, we talked about what chromatin is, right? So again, it's a point of confusion. Chromatin, right? It's DNA plus what? What's chromatin? It's not just DNA, it's DNA plus.
Protein where it surrounds it.
Right, what you call?
I think it begins with an H.
Histone, yes. So chromatin is DNA plus histones, plus there's some other proteins that help with the packaging, right? But we're not going to worry about this. It's mostly the DNA plus the histones. And why is that so important? Because remember, again, if you think about it, you have a piece of string, you're like, well, it's a shame to throw this piece of string away. you'll just find like something and wrap it around it and, you know, like maybe you'll use it later, right? So that's a way of organizing things. So the chromatin has DNA plus protein. That's a way of helping us organize and condense the DNA. They condense down into chromosomes that you can actually see, right, even with a light microscope, and they become visible, right? So this is the only point when they become visible is like sort of late prophase and into metaphase, right? Each is composed of two sister chromatids held together by a centromere, right? We talked about this. That's the protein that was holding the sister chromatids together. And then this thing called the spindle starts to form. The spindle is actually formed at the two ends of the two poles of the cell. What's happened is the centrosomes have migrated to the two ends of the cell. And then this football-shaped spindle apparatus, which is very three-dimensional with all these microtubules going back between the two centrosomes is going to form. Okay, then we go into pro-metaphase. Now, what we need to do is organize the DNA so that's eventually in metaphase, all the DNA is going to be lined up right in the middle of the cell. You guys remember that picture from the video? So how do we get there? Well, we're in pro-metaphase, which is right before that. So if you look at your sister chromatids, and for illustration purposes, I removed the centromere, but it's still there. I didn't want to complicate things too much. And what I've done is I've labeled this picture. We have the sister chromatid. We have the two centrosomes, which are polar opposite the cell ends of the cell now. We have microtubules, which are these also referred to as the spindle fibers. They're reaching out and they're actually contacting the DNA. The sister chromatids are what are called kinetochores. So kinetochores, well, each kinetochore is made out of protein. Okay, so it's like an anchoring spot for the spindle fiber, which is a microtubule to then attach. Okay, so kinetochores appear on each side of the centromere, again, which is not shown here. The kinetochore of each chromatid is attached to a kinetochore spindle fiber. So this microtubule is often referred to as a kinetochore spindle fiber because not all spindle fibers connect to kinetochores, right? Just some do. So that's why this is called a kinetochore spindle fiber. The spindle fibers extend from the poles to the chromosomes. So the poles of the cell, like sort of football shaped, right? And they're going out to the middle, and you have the chromosome sort of in the middle there. The kinetochores pull the chromosomes back and forth toward alternate poles to begin aligning the chromosomes. So this is actually a very interesting process, which often isn't shown, but the chromosomes, to get them in the middle, there's actually kind of this tug of war where the kinetochores will pull one way, and then they'll pull the other way, and back and forth and back and forth. And eventually, through this process, it eventually gets all of them lined up in the very middle. And that's actually metaphase, once all these chromosomes are lined in the very middle. Okay, so let's take a look at a picture of this. Here we've got a cell, animal cell interphase. And so we've already had the S phase. Here we have early prophase. So what you can see here are the centrioles, right? Within each centrosome. And you see the word aster, that aster literally means star. So what you can see is it looks like a star because all the microtubules are sort of emanating out of it, right? It's sort of like a star. So now one of these centrosomes with its centrioles is going to end up migrating to the other side of the cell. Nuclear envelope starts to fragment. We don't need it anymore. The nucleolus disappears. That's again for the assembly of ribosome subunits. Chromatin condenses. We talked about how that happens, right? The centrosomes the centrosomes duplicate, right? So normally you just have one centrosome. Now you have two. Okay, now the spindle is starting to form as these things. This one is going to migrate all the way. This centrosome is going to migrate all the way to the opposite pole of the cell. Now into prophase, the nucleolus has disappeared. The duplicated chromosomes are now visible because they've condensed so much, right? So they were there, they just couldn't see them before, but they weren't condensed enough. The centrosomes begin moving apart. Spindles in the process of forming. So you can see this here in the real picture. This, I think, yeah, probably a fish cell, but I'm not for sure. Often they use a fish cell for this. Yeah, you can see here an aster right there, right? So there's a centrosome. Here you can see two, right? And you can see the chromosomes are much more condensed here, and they're starting to get closer to the center. Here's pro-metaphase. So we're getting closer to metaphase. So that, remember, there's a tug of war now. These kinetochore spindle fibers are pulling one way, then the other, and they're getting the chromosomes to line up in the very middle. The kinetochore of each chromatid is attached to the kinetochore spindle fiber. The polar spindle fibers stretch from each polar spindle pole and overlap. So now we're starting to get, you see these, it's starting to form like kind of a football shape because they're sending out these fibers and they're actually starting to overlap. You can see the sort of structure forming here. All right, so now we're on to metaphase. So the spindle is fully formed in metaphase and consists of poles, asters. Remember, these are all these microtubules. They look like rays from a star. That's why they're called asters. And fibers. The polar fibers are overlapping. We saw that a second ago. The centromeres of chromosomes are aligned. Oh, the centromeres. So now we're talking about the centromeres. So remember the centromeres. Let me redraw this. I'm just going to do a single chromosome. Okay, so we have a blue. Hold on, sorry. We have sister chromatids now, right? So these are sister chromatids. That are shown here. There's a centromere that's holding them together. There's the kinetochore protein. They're kind of like oval discs of protein. What's attached to that? We have the microtubules attached here, right? That's what we have right here, okay? So the centromeres are aligned at the metaphase plate. Then after that, we have anaphase. The centromeres divide. So this protein, which is a centromere, divides. And so that was holding these sister chromatids together, right? So now, remember these kinetochore spindle fibers were pulling like this in this direction, right? Towards the opposite pole. So what will happen in anaphase? As soon as this, as soon as that disintegrates, that centromere, suddenly they're going to split very quickly, right? They're sort of, they go towards the opposite poles. The kinetochore spindle fibers shorten, pulling the daughter chromosomes. The spindle fibers, the polar spindle fibers push the poles apart. And we continue to push the poles apart. Yeah, go ahead.
So do the split bring like birth defect, or is it the lack of the protein?
Oh yeah, so we're going to talk about that later. There's we're going to get to that. It's a whole topic in itself, so we'll get to that later. Okay, but yeah, things can happen wrong that can lead to things like Down syndrome and things like that. Yeah. So the next phase is the last. Well, yeah, it's one of the last phases, which is telophase, where the spindle is going to disappear now. We don't need it anymore. We can use that. We can reuse those microtubules for the cytoskeleton of the cell of the daughter cell. The nuclear membrane components reassemble around the daughter chromosome. So we need to make that double membrane nuclear membrane again with the nuclear pores and so forth. Chromosomes become diffuse again. So we don't want our chromosomes when you know all condensed, because they can't do anything there. Like they can't direct protein synthesis and direct the cell, right? Because the DNA really, in the sense, is the controlling molecule of the cell. The nucleolus starts to appear because we're going to need to start making ribosomes again, right? And then cytokinesis begins. Okay. So let's take a look at some pictures for this. So here we are in metaphase. So we have what's called a metaphase plate where everything's lined up really nicely, right? Almost perfectly, really, right? And you have the sister chromatids, right? Like what I've drawn here. So that's actually considered like a single chromosome there, right? But now as soon as they split, this goes from one chromosome, it's now two chromosomes, right? But the chromosomes have half the amount of DNA now because each one is just an individual chromatid, right? Same thing, so this is for the short one, here's a long one, here's a long one, here's a short one, right? So divided up, and then eventually, so you see the nucleus starting, so that's then anaphase, and then here's telophase over here. The DNA is in fact now split up, and now we have the nuclei starting to form again, and we see a cleavage furrow begin to occur here. So, yeah, the cells elongate, but then what happens is it's a lot like you probably have, you might have at some point in your life had a little purse with a string that closes it, right? You know, like a purse where you put coins or something like that. You have a string that you pull on the string that tightens the mouth of the purse. That's essentially what happens here. We have microfilaments, which are made out of a protein called actin. So actin is a very important protein. We're going to talk a lot about it. It's found in your muscles and so forth. They make a circle around here, and they got tighter and tighter and tighter and tighter in animal cells. And it starts with a little furrow, but eventually it completely splits off the two cells. Okay, so it's a lot like closing the top of a purse where you might hold coins or something like that with a string. Okay, so that's kind of that. Okay, so let's take a look at mitosis in plants. Of course, plants are very important, so we need to talk about them at least a little bit. So they have to do the same thing. They have to develop, they have to grow, they have to repair, and so forth. So in plants, a lot of the mitosis occurs in what are called meristematic tissue. These can divide throughout the life of a plant. So here, for example, we have a plant, and it has a terminal bud. You see that at the very top. So the terminal bud has this very delicate tissue at the very end. And if you zoom into it, you can see what's called an apical meristem. Apical just means at the apex of something, but the top. So these are actively dividing cells. They're essentially stem cells. They're dividing and making new cells. And what they're doing is they're making these leaf primordial, which is essentially baby leaves, right? So making little baby leaves will become bigger and bigger, and they'll continue to grow and push upwards. So this plant will get bigger and bigger, maybe turn into a tree or something like that, or just be a bigger plant. Now, the tree also has to get, or a bush has to get wider in diameter, right? For its branches and so forth. So it has, within these woody plants, you're going to have a vascular cambium and a cork cambium. So the cork cambium is another layer of meristematic tissue that can divide throughout the life of the plant. And it's going to make essentially the bark. It's going to make the cork or the bark out, which is going to protect the cells, like the epidermis of a tree or a plant. And then there's the vascular cambium. It's going to do everything else. So it's going to make, this becomes like a mini little tree or something. It's making all that material that's going to form the bulk of the tree, the inside of the tree, right? All that. Okay, so that's the two types of cambium. Oh, there's another one too. If you look at the roots, we have a root apical meristem. So it's right here, and so it's very actively dividing, pushing the root further and further deeper into the soil. Now, this tissue is very sensitive, so it actually makes sense for the root. The apical meristem is not directly in contact with the soil, right? Because it could get damaged. So there's a root cap which is out in front, which allows the root to sort of explore, look for nutrients and water and so forth. Okay. So plants essentially go through all the same phases as animal cells. One of the big differences is that the majority of plants that you see out there do not have centrioles or afters. Okay, some do, but most don't. So here's an example where we have a plant cell. This one actually does have centrosomes, but no centrioles. So here's one that has centrosomes but lacks centrioles. So again, this is a little unusual, but some plant cells do have this. So in early prophase, the centrosomes are duplicated. So chromatin is condensing into chromosome. The nuclear envelope is fragmenting. In prophase, the nucleolus has disappeared. The duplicated chromosomes are now visible. You can see this here. The chromosomes will begin moving apart, and the spindle is in the process of forming. So they do have a spindle. So they do have a spindle made out of tubulin, just like an animal cell. It's just organized a slightly different way than in animals. Pro-metaphase, we're getting close to metaphase. The kinetochord or each chromatid is attached to the kinetochord spindle fiber. The polar spindle fibers stretch from each spindle pole and overlap. So we're getting these Here's a spindle pole, but there is no centriole. No centrioles and no aster. Metaphase, everything's lining up. We have the metaphase plate. Centromeres and duplicated chromosomes are aligned to the metaphase plate. Kinetic or spindle fibers attached to the sister chromatids. They come from the opposite pole of the cell, the spindle poles. Anaphase, the chromosomes are now pulled apart because the centromere disintegrated, just like in animal cells. The chromosomes move apart toward the spindle poles, and in this way, each pole receives the same number and kinds of chromosomes as the parent cell. And then we have telophase. The daughter cells are forming as nuclear envelopes and nucleoli reappear. Chromosomes will become indistinct chromatin again. So here's a picture here.
In animal
Just
cells,
cases in animal
cytokinesis
cells.
completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells.
Yeah, so that's the big difference. So in animal cells, here's an animal cell. You can see the cleavage furrow. This is because the actin fibers are pulling. And they're going to get tighter and tighter and tighter, and then they're going to even have two daughter cells. So you always start with the cleavage furrows. In plants, you have a cell plate. It starts with these vesicles forming. And remember, because most plant cells have a cellulose wall around them, right? So eventually you'll have this cell plate, which is pretty rigid, and it's going to start forming. It's going to have, eventually you'll have a full cellulose wall between the two cells, okay? So quite different, but it makes sense given how different plant cells are from animal cells. All right, so that's it for mitosis. Any questions on that? There's a lot of material. Your book is pretty good, by the way. So if you go through and carefully read through the book, you'll understand everything pretty well. It's a lot. There's a lot to take in at once. So we're going to just, I have only a couple of minutes, but we'll at least get an introduction to meiosis, which again, this is very, I actually recommend reading this because it's mitosis is easy. That's why we do it first. It's easier than meiosis. Meiosis gives students a lot more trouble because it's a little more complex process. All right, but let's just get into what meiosis is. And again, I recommend you go to your textbook and do a little reading. We'll go over meiosis next time. But meiosis, unlike mitosis, occurs only in reproductive cells. So the germline, the cells, it's essentially a process for making eggs and sperm cells. In this process, the amount of genetic material in the germline, which are going to be diploid, is reduced by 1/2 to produce gametes. So gametes will be haploid. The cells that produce these haploid cells are in fact diploid, right? And we'll talk about that in more detail later. So what are the gametes? Well, there's just two types of gametes. There's sperm cells. There's the male gamete. It's just the male gamete. It's relatively small cell made by males. There's the egg cell. This is a bigger cell. This is the female gamete produced only in females. And really, that's kind of the big distinction. The sperm cell is usually smaller than the egg cell. That's when we're distinguishing. The sperm cell is often more motile, meaning it can move more than the egg cell. Okay, but we'll get into that later. So each gamete contains half the number of chromosomes found in all somatic cells. So it's haploid. Remember, another way of saying haploid is, which means single in Greek, is using the abbreviation N. So it just means that we have the N number of chromosomes. And then once you go through fertilization, you're going to make a new individual, right? That's called the zygote. So that all of you were a zygote. You're all a single-celled organism at one point. And that has to have the diploid number, right? So that's the double number. Again, diploid means double in Greek, so it's the 2N number. So that's the full complement of genes. So again, in us, we have 23 pairs or 46 chromosomes, right? So our cells, all our somatic cells, have 46 chromosomes, but something like a sperm or an egg cell would only have 23. So what does this look like? Well, you have the haploid sperm, so it has the N number of chromosomes. And here we simplified it, right? Instead of showing all the, it would be 23, right? Obviously in a human, but we're just showing 3 here. And here is a haploid egg cell, we're just showing 3 here. And they get together essentially to form this fertilized egg, which is the diploid zygote. It goes back to having the 2n number of cells. Right. So, and you can see what they're showing you here is the paternal genes are all in blue. And then the maternal genes are all shown, chromosomes are all shown in green, right? So those are going to form homologous pairs of chromosomes when they get together. So, like mitosis, meiosis is preceded by the replication of chromosomes during the S phase or interphase. And we're going to talk about next time the fact that meiosis really has 2 stages. There's meiosis 1 and meiosis 2. Unlike meiosis, the 2 cell divisions in meiosis result in 4 haploid daughter cells. And we'll talk about that next time. Each daughter cell has only half as many chromosomes as the parent germline cell. And then meiosis 2 is finally completed actually only after fertilization, when the sperm actually fertilizes the egg. Okay, so we'll stop there and we'll pick it up with meiosis. So again, I recommend, because of how confusing meiosis is, you may want to read up on it. Okay. All right, thanks guys, and see you next time.